And I gotta find a better picture of this, but I like this in general because it shows general properties between the or different types of lipoproteins. So you might have heard of things like LDL, HDL, and sometimes IDL or VLDL. So what they are basically phospholipids containing different balances of proteins, triglycerides, and cholesterol. So this is why I kind of like this, very simple, but I think it does a neat job of showing relative amounts of these proteins and triglycerides and cholesterol. So what do they have all in common? They're hydrophilic on the outside. And why? Because you want to be able to transport these lipids in your blood without your lipids gumming up the rest of your circulatory system. But they have that hydrophobic inside. And why? Well, where are they transporting? They're transporting triglycerides and cholesterol. And so what we have here, so the liver is going to make liver is going to make bile salts. That's going to break up dietary fats into micelles. Your enteroendocrine cells are going to, or actually lipases are going to break apart the micelles and make free fatty acids and monoglycerides. Your enteroendocrine cells are going to absorb these free fatty acids and monoglycerides, repackage them into chylomicrons. These go back to your liver, which repackages them into lipoproteins. And then these lipoproteins are going to go to the rest of your body. And there are different types of lipoproteins. Don't worry too much about the details here, but this is what I'm getting at here. So again, your liver is a big repackaging warehouse for lipids in your body. And it can secrete things like LDL and VLDL. Now this is an interesting thing too, is like these peripheral cells or tissues of your body, this is where, so the way you get lipids from your liver to other tissues in your body are VLDLs and LDLs. Now your tissues of your body, they can actually take these LDLs, repackage them further, and then make them into HDL parcels. Now, this is where it's kind of controversy because now we're finding, we used to think like, okay, you might have heard of things in, like if you've been to the doctor's office or have shadowed anyone in the clinic, you may have heard like, oh, good cholesterol and bad cholesterol. So they used to think HDL is the good cholesterol and LDL is the bad cholesterol, because like high levels of LDL, well, LDL is always going to be like high levels of LDL, that's still unhealthy. Like it predisposes you to cardiovascular diseases, especially cardiovascular disease. That's one of the big things that you have to watch out with high LDL. They used to think like, oh, maybe a high HDL offsets a high LDL and high cholesterol, but now they're finding like maybe high HDL isn't too good as well. It's not necessarily associated with longer life. So the mnemonic that used to be helpful was H is healthy, L is lousy. But L is still lousy, but now whether HDL, high density lipoproteins, are still healthy, that's kind of like something that might change it within the next maybe decade or so. But what's happening again? So different ways lipoproteins transport lipids in your body. That's what I'm getting. That's one of the main takeaways from that image. Now there's a condition called hepatic steatosis, aka fatty liver. So this is what a normal liver looks like, and notice it has kind of a reddish brownish kind of healthy looking color. So this is what a healthy liver looks like. It has this kind of dark red color. Or if you've ever gone to the meat, I don't see it too often, but you can probably go to the butcher and see what a liver looks like. This is a healthy color. Now this is a liver that's cross section, but notice that it's kind of like off colored, right? It's kind of a creamy yellow off-white color, and it's actually a little bigger too in terms of thickness. So this liver has a lot of fat content. So this is what hepatic steatosis is, also what we call fatty liver disease. And what we have here is that here on this picture we have a histology slide. This is what a normal cross section of hepatocytes and lobules should look like. But over here, notice that we have all these big pockets. So when you take a tissue section and do standard staining in it, you wash away all the lipids as well. So all these little pockets right here in the sample from hepatic steatosis or fatty liver sample, these used to be filled with triglycerides and lipids. It was just washed away during the whole fixing and staining process on the microscope slide. And if you've eaten this, I mean, what this is foie gras, which is literally fat liver in French. And notice which color it looks like. It looks more like this color than the previous color, right? So again, this is why the fatty liver is, this is what happens with the foie gras, is you overfeed a duck or goose, and then they end up with hepatic steatosis. That's why foie gras tastes like that. That's why someone called it like duck butter, because it's basically all the fats in the liver, and that's what you're eating when you eat foie gras.